All right, let's dive into one of the most fundamental topics in all of pathology. And I'll tell you right now, this is a guaranteed question on your oral exams: how cells respond to stress. If you can understand this whole sequence from adaptation all the way to death, you'll have the key to interpreting pretty much every biopsy slide you ever see. Let's get into it.
So what happens? When a cell gets hit with some kind of stressor, toxins, an infection, or just chronic irritation, it doesn't just keel over and die immediately. No, it's got a whole playbook of responses. And those responses are exactly what we as pathologists are looking for to figure out just how bad the injury really is.
You can think of it like a three-act journey for every single stressed cell. First, it tries to adapt. It changes to survive. If that stress just keeps on coming, it can become disordered. And now it's walking a very dangerous line, a precancerous state. And finally, if the injury is just too much, it hits the point of no return, death. So let's break down each one of these stages. Okay, so our story starts with adaptation. This is the cell's survival playbook, its first attempt to find a new kind of normal, even in a hostile environment. Before any of the drama starts, our cells are living in this happy, balanced state called homeostasis. It's this perfect equilibrium between new cells being made and old cells being lost. Stress is the thing that comes along and knocks that whole system off balance, forcing the cell to figure out a new plan. Now, how does a tissue adapt when it has to handle an increased workload? Well, it's got two main strategies. The first one is hypertrophy. That's an increase in the number of cells. The classic example is the liver's amazing ability to grow back. The second strategy is hypertrophy, and that's where the cells you already have just get bigger. Think about a bodybuilder's muscles, so more cells versus bigger cells. It sounds simple, but that distinction is absolutely critical. And here is the key rule you have to remember for your exams. Hyperplasia can only happen in cells that are actually capable of dividing, like the cells in the endometrial lining. Hypertrophy, on the other hand, is the only option for permanent cells that cannot divide, like your heart muscle cells trying to cope with high blood pressure. So the cell's ability to divide, or not, is the deciding factor. But, adaptation isn't always just about size or number. Sometimes cells do something really clever called metaplasia. This is a reversible change where one specialized cell type gets replaced by another one that's tougher and better suited to the stress. It's a smart survival move, but it's a trade-off. The new cells might be more durable, but they often can't do the original job. And the classic super high-yield example of this is Barrett's esophagus. Here, the stressor is chronic acid reflux. In response, the delicate, flat squamous cells of the esophagus literally change into the tough, mucus-producing columnar cells you'd normally find in your intestine. They survive the acid way better, but this new identity, well, it can set the stage for bigger problems down the line. Now, if that stress that caused the metaplasia in the first place just keeps going, we enter the danger zone, dysplasia. And let me be clear, this is no longer a true adaptation. This is a big step towards neoplasia, towards cancer. Dysplasia literally just means disordered growth. When we look under the microscope, we see that the cells have lost their normal, uniform look and their organized structure. We're looking for things like pleomorphism, which is just variation in cell size and shape, and dark, angry-looking nuclei. This is a precancerous lesion, and it's basically the last exit on the highway before malignancy. Okay, this is a key comparison for your exams. You need to know the difference. Metaplasia is an orderly change from one cell type to another. Dysplasia is disorderly, chaotic growth of abnormal-looking cells. And while metaplasia can set the stage for cancer, dysplasia has a high risk of progressing to it. Getting this distinction right is absolutely fundamental. And that brings us to the final act. When the injury is just too severe or goes on for too long, adaptation and even dysplasia fail. The cell has to die. But how a cell dies is every bit as important as why. Does it go out with a bang, a messy, inflammatory explosion that damages all its neighbors? Or does it undergo a silent, pre-programmed controlled demolition where it gets neatly cleaned up without a trace? That explosion, that's necrosis. This is always a pathological process, always caused by some kind of irreversible injury. The cell membrane just falls apart, its contents spill everywhere, and this whole mess triggers a massive inflammatory response. Think of it as a ton of collateral damage. Now, necrosis shows up in a few different ways. In coagulative necrosis, which you see in a heart attack, the basic tissue structure is still there, but the cells are just these pink ghosts with no nuclei. In liquefactive necrosis, like in a brain infarct, the tissue basically turns to soup. And in caseous necrosis, the hallmark of tuberculosis, you find this crumbly, white, cheese-like debris. Each pattern points you toward a different cause.
The silent demolition, on the other hand, is apoptosis. This is programmed cell death. It's an active process. It actually costs the cell energy to do it. But it's clean, it's controlled, and it does not trigger inflammation. It's actually essential for normal things like development and just getting rid of old, worn-out cells. So here it is, the big showdown. Necrosis is passive, it's messy, and it is always inflammatory because the cell swells up and bursts. Apoptosis is active, it's clean, and it's non-inflammatory because the cell shrinks down and neatly breaks apart. You can think of it this way. One is murder, the other is suicide. So how does this controlled demolition actually happen? Well, the main pathway is regulated by a family of proteins. When a cell gets stressed, pro-death proteins like BAX and BAC punch holes in the mitochondrial membrane. This lets a molecule called cytochrome C leak out, and that is the point of no return. Cytochrome C kicks off a chain reaction that activates the initiator caspases, which then activate the executioner caspases. And these executioners, they're the demolition crew. They go to work systematically chopping up the cell's skeleton and its DNA. Okay, let's pull all of this together. If a professor asks you to summarize cellular injury in an oral exam, how do you give them that perfect, concise answer? Here's your 2-minute oral exam answer. You'd say, Professor, cells first respond to stress by adapting. This can be through hypertrophy, which is an increase in cell size, like in cardiac muscle, hyperplasia, an increase in cell number, like in the endometrium, or metaplasia, a change in cell type, like in Barrett's esophagus. If the stressor doesn't go away, this can progress to dysplasia, which is disordered, pre-malignant growth, and finally, if the injury becomes irreversible, the cell will die, either by messy, inflammatory necrosis or by clean, programmed apoptosis.
All right, let's try a quick practice question. A biopsy from the airway of a chronic smoker shows that the normal columnar cells have been replaced by tough, flat squamous cells, which are better at handling the smoke. What do we call this adaptive change? And the answer is B, squamous metaplasia. It is a perfect example of one mature cell type swapping itself out for another, more resilient one because of chronic stress. So why does all of this matter so much? Because this isn't just theory. This sequence, from adaptation to dysplasia and potentially to cancer, is the absolute foundation of pathology. It's how we read biopsies, it's how we understand how diseases progress, and yeah, it is absolutely 100% guaranteed to be on your exams. And that leaves us with one final thought to chew on. We've seen that both life and death are quite literally programmed into our cells. The real question is, what could we achieve if we truly learned how to control that switch? Could we stop cancer in its tracks by flipping on apoptosis, or maybe halt neurodegeneration by preventing it? The possibilities are as profound as the process itself.